All right, let's begin with the evidence. Number 10. The cave floor with no bodies. At True Al West Cave in Belgium, one Pleistocene layer contained no human bone, no tooth, no skull, no fragment that anyone could identify as human. Yet when researchers tested the soil, they found damaged Neanderthal DNA. For a long time, archaeology worked by a simple rule: no body, no proof that people had been there. This cave challenged that rule. An extinct human group had disappeared so completely from the visible record that the only thing left to remember them was the floor beneath the cave. In 2017, Vivian Sloan's team screened sediment from 4 Eurasian caves and recovered Neanderthal DNA in 8 archaeological layers. At Denisova Cave in Siberia, they also found Denisovan DNA in a much older layer. The conclusion was both simple and unsettling. Ancient DNA could reveal people who had left no recognizable body behind. How many human groups vanished from history because their bones never survived? That question changes the way a cave must be understood. It is no longer just a shelter full of tools, ash, and animal remains. It can also be a quiet record of people who entered its darkness, left traces of themselves, and disappeared until a few molecules in the soil brought them back into view. The method depended on searching for tiny fragments of hominin DNA preserved in the sediment itself. Researchers used targeted enrichment, a laboratory technique designed to isolate molecules most likely to come from ancient humans from the overwhelming background of animal, plant, fungal, and modern DNA. The science is technical. The implication is not. Archaeologists no longer had to wait for a skull, a tooth, or a recognizable bone before asking who had occupied a cave. How can a human presence survive after the human body itself is gone? In the first study, the answer came from carefully sampled layers at 7 sites. Neanderthal DNA appeared in sediments from 4 of them, including layers where no hominin skeletal remains had been found. At Denisova Cave, the same method recovered Denisovan DNA from a layer near the bottom of the sequence. Empty ground had taken on a different meaning. What once looked like background dirt could preserve the last biological trace of people who had made fires, processed food, rested in shelter, or simply passed through without leaving anything visible behind. Later work showed how much further the method could reach. In 2021, researchers analyzed 728 sediment samples from Denisova Cave. Those samples traced changing evidence of Denisovans, Neanderthals, and later modern humans across more than 300,000 years of occupation. The cave was no longer defined only by a few famous bones. Its floor became a map of arrivals, absences, and returns. It also allowed archaeologists to compare DNA signals with stone tools, animal bones, and changing climate conditions that can place a vanished population within a particular moment of a cave's history, even when the people themselves are missing. Soil does not become a complete diary simply because it contains DNA. Sediment DNA is fragmented, damaged, and vulnerable to contamination. A positive genetic signal can show that a population was present in a layer. It cannot identify a face, recover a name, or tell us whether the people who left that DNA were hunting, sleeping, grieving, or preparing to leave. Still, the central conclusion stands. Ancient DNA made it possible to find extinct humans in places where their bones had disappeared entirely. It gave archaeology a way to recognize a presence that had once been invisible, not through legend or speculation, but through damaged molecules preserved beneath a cave floor. That changes the scale of loss. A population may leave no grave, no complete body, and no tool that can be assigned to them with certainty. Yet a few fragments of DNA can still show that they were there. The next evidence file moves from invisible people in soil to one child preserved in bone. A girl from Denisova Cave whose mother was Neanderthal and whose father was Denisovan. Number 9. The girl with 2 extinct parents. The fragment was only a small piece of a long bone. It came from Denisova Cave, found among more than 2,000 bone, fragments that could not be identified by eye. Nothing about it initially suggested a family. History no living person could have imagined. Collagen fingerprinting identified it as hominin. Mitochondrial DNA placed it on the Neanderthal side of the ancient human family. Direct dating showed that the bone was more than 50,000 years old. Its thickness suggested that its owner had been at least 13 when she died. Then the nuclear genome produced a result that went far beyond a simple label. About half of the informative genetic markers matched Neanderthal DNA. The other half matched Denisovan DNA. The pattern ran evenly across her chromosomes, exactly as researchers would expect in a first-generation child whose parents belonged to two different populations. Her mother was Neanderthal. Her father was Denisovan. The girl is known as Denisova 11. Neanderthals and Denisovans had diverged more than 390,000 years before she was born. For a long time, textbooks drew them as separate branches, one associated mainly with Western Eurasia, the other known from a few bones in Siberia, and a genetic legacy scattered across parts of Asia and Oceania. Denisova 11 made that picture feel far less settled. How often did people from those populations meet closely enough to form families? Her DNA could not answer every part of that question, but it provided much more than one dramatic fact. The girl's mitochondrial DNA came from her mother, confirming the Neanderthal line. Her X chromosome showed that she was female, and the way Neanderthal-like and Denisovan-like genetic variants were distributed across her genome showed that she was not. Descended from a population mixed for countless generations. She was the direct daughter of one Neanderthal parent and one Denisovan parent. The result offered a clear payoff. The boundary between these extinct populations was real, but it was not absolute. Her family history contained another complication. The Neanderthal ancestry in Denisova 11 was more closely related to later Neanderthals in Europe than to an earlier Neanderthal found in the same Siberian cave. Her Denisovan father also carried traces of Neanderthal ancestry. Movement and mixing, then, were already shaping both sides of her family long before she was born. The cave had not held two isolated populations, each confined to its own corner of prehistory. It formed part of a wider human landscape, connected by migrations across Eurasia that archaeology can still trace only in fragments. That does not mean Neanderthals and Denisovans lived as one population. Their genomes remain distinct. Their zones of contact may have been narrow in time and space. The bone cannot tell us whether her parents met through travel, exchange, seasonal movement, conflict, adoption, or a relationship that left no other trace. It does tell us that the encounter happened, and it may not have been rare. Of the 6 individuals from Denisova Cave for whom nuclear DNA was available when the study was published, 2 showed evidence of mixing between Neanderthals and Denisovans. That is too small a sample to measure how often. Such relationships occurred across all of Eurasia. But it makes the old image of separate human populations, passing one another without contact, much harder to keep. Denisova 11 was not a theory about interbreeding. She was the physical result of it. One small bone fragment preserved proof that 2 extinct populations, separated for hundreds of thousands of years, could still become one child. The next case carries that question out of Siberia and into a Romanian cave, where a modern human preserved the genetic trace of a Neanderthal relative close enough to sit within a family memory. Number 8. The man whose ancestor was Neanderthal. A human jawbone lay deep inside Peștera cu Oase, the cave with bones, in southwestern Romania. There was no face left to reconstruct, no grave, no tools, and no account of the people who had carried it into the darkness. Yet inside that bone, the DNA of a man who lived roughly 37,000 to 42,000 years ago preserved. A family connection that should have vanished with the Neanderthals. His genome held far more Neanderthal ancestry than scientists expected in an early modern human. Most present-day people with ancestry outside sub-Saharan Africa carry around 1 to 3%. Oase 1 carried roughly 6 to 9%. More importantly, several of those Neanderthal segments were still exceptionally long. That detail changed the story. DNA is reshuffled every time it passes from parent to child. Over enough generations, inherited segments become shorter and more scattered across the genome. Oase 1's Neanderthal DNA had not yet been broken apart in that way. So how close was that Neanderthal ancestor to the man whose jaw lay in the cave? Close enough to make prehistory feel uncomfortably recent. The size of those segments suggested that Oase 1 had a Neanderthal ancestor only 4 to 6 generations earlier. In ordinary family terms, that could mean a great-great-grandparent, or someone only a little further back. The DNA cannot tell us what kind of encounter brought the two populations together. It cannot show whether people met through seasonal travel, shared territory, exchange, coercion, adoption, or a relationship that left no memory beyond a chromosome. It does, however, settle something more basic. Neanderthals and early modern humans in Europe were not merely replacing one another from a distance. At least sometimes they lived close enough for their families to overlap. The jawbone came from a period near the end of Neanderthal existence in Europe. Neanderthals are generally thought to have disappeared there around 39,000 to 41,000 years ago. Oase 1 lived during that final stretch when modern human groups were entering territory Neanderthals had occupied for hundreds of thousands of years. The older story made the transition look clean. One population arrived, another faded, and Europe became modern human. Oase 1 made that boundary harder to draw. A Neanderthal relative was not buried deep in some remote evolutionary past. That person may have stood within the living memory of Oase 1's family. The genetic evidence carried a darker result as well. Oase 1 does not appear to share more genetic ancestry with later Europeans than with East Asians. The most likely interpretation is that he belonged to an early modern human population that entered Europe, mixed with Neanderthals, and then did not contribute substantially to later European populations. His story, therefore, contains two disappearances. The Neanderthals and his family were nearing the end of their existence as a distinct population. Oase 1's wider population may also have faded from Europe's later genetic history. That is the payoff of the jawbone. Ancient DNA did not simply confirm that Neanderthals and modern humans mixed. It revealed a man standing at the edge of two disappearing worlds. One population already vanishing, another leaving little trace in the people who came after. The limits of the evidence still matter. A few long DNA segments cannot become a complete family story. The cave did not preserve a name, a camp, or the circumstances of a relationship. But it preserved enough to show that extinction is messier than the diagram suggests. A people can disappear as a separate population, while part of them survives in another family. A family can exist where two human histories meet, then vanish so completely that one jawbone becomes the only witness. The next file widens that intimate discovery into something larger. Several early modern humans in a Bulgarian cave, each carrying evidence of a recent Neanderthal relative, and each belonging to a population later Europe almost entirely forgot. Number 7. The first Europeans who left no Europeans behind. At Bacho Kiro cave in Bulgaria, the evidence began as scraps, a tooth and bone fragments mixed among thousands of animal remains. They came from people who entered Europe more than 45,000 years ago, while Neanderthals still occupied parts of the continent. Researchers could not identify the fragments by shape. Too little remained. Instead, they used collagen peptide analysis to separate human bone from animal bone, then directly dated the remains and their layers. The result placed modern humans in the cave during the initial Upper Paleolithic, among the earliest known Homo sapiens in Europe. That already mattered. Europe had not simply changed hands after Neanderthals vanished. Modern humans were moving through the same landscapes, using some of the same caves, and leaving tools during the same period. The genomes made the encounter far more personal. Several Bacho Kiro individuals carried unusually long stretches of Neanderthal DNA. Those stretches mattered, because inherited DNA is reshuffled with every generation. Over time, long pieces are broken into smaller ones. In these people, the segments were still large enough to show that a Neanderthal ancestor stood only about 5 to 7 generations back. Why did some of the first modern humans in Europe carry Neanderthal relatives close enough to sit inside a remembered family past, yet leave so little clear ancestry in the Europeans who came later? The first answer lay in the DNA itself. The Bacho Kiro genomes did not fit neatly with the later populations that would form much of Europe's genetic history. They showed closer connections to ancient and present-day populations in East Asia and the Americas than to later Western Eurasian Europeans. The people at Bacho Kiro belonged to an earlier movement of Homo sapiens into the continent. They met Neanderthals often enough for recent ancestry to appear in several genomes. They left stone tools, broken bones, and genetic evidence of families formed at the edge of two human worlds. Yet their own population did not become the main ancestry of later Europeans. The payoff is clear. Ancient DNA revealed that some of the earliest modern people Europe were not the beginning of a continuous European population. They were part of an early human movement whose genetic path largely faded from the region. That does not mean every person at Bacho Kiro vanished without descendants. A handful of genomes cannot trace every family. Through 45,000 years of migration, absorption, population loss, and change. But the broader pattern is difficult to ignore. Europe was entered more than once by populations whose arrival was real, whose contact with Neanderthals was real, and whose distinct genetic signatures later grew faint or disappeared. The cave makes that loss feel physical. A few teeth and bone fragments lay in darkness long after the people who carried them had vanished from Europe's later family tree. There were no names, written accounts, or grave goods explaining where they came from or where their descendants went. There was only evidence of a group that reached Europe early, lived close to Neanderthals, and then passed out of the genetic story that followed. Their disappearance also changes the old idea of replacement. Human history did not move in a clean sequence in which one population arrived, another left, and the map settled into something familiar. It was crowded. Small groups crossed unfamiliar ground, met other humans, formed families, split apart, and sometimes disappeared before their presence became the ancestry of anyone living later. Bacho Kiro offers a glimpse of one such population at the moment it became visible again. Not because it left a city or a monument, because a few broken bones still carried the memory of people Europe almost forgot. The next case leaves Europe for Ice Age Siberia, where a child's genome revealed a population that vanished as a separate people, but still survives in the ancestry of communities on two continents. Number 6. The boy who connected two continents. At Malta, in south-central Siberia, archaeologists uncovered the remains of a young boy who had lived around 24,000 years ago. His bones lay in an Ice Age landscape of long winters, open steppe, and distances that could turn a missed hunt into a threat to an entire family. The site had already produced stone tools, worked mammoth ivory, and traces of people surviving far from any easy source of warmth. Yet the boy, known as MA1, initially seemed to be one more fragment from a remote Upper Paleolithic camp. His genome revealed something much larger. When researchers sequenced MA1's DNA in 2014, they found that he was genetically connected to Western Eurasians and to Native Americans, while showing no close affinity to East Asians. He belonged to a population now described as Ancient North Eurasian, a population that no longer exists as a separate people. How could a child who lived deep in Ice Age Siberia be genetically tied to people on two distant continents, yet not fit neatly into the modern populations surrounding his homeland? The answer changed the older map of northern Eurasia. For a long time, the story of the first Americans was reduced to a simple movement. People from East Asia crossed Beringia and entered the Americas. The broad movement remains real, but MA1 showed that the ancestry behind it was more complicated. The first American populations did not emerge from one ancestral source alone. They carried ancestry related both to East Asian groups and to a population related to the boy from Malta. That did not make MA1 a direct ancestor of every Native American community, and it does not mean his own family crossed into the Americas. DNA cannot follow one child's descendants through every generation. It can, however, identify the wider population history around him. The study estimated that roughly 14 to 38% of Native American ancestry may have come from a population related to MA1. That ancestry likely entered the ancestors of the first Americans after their separation from East Asian ancestors, but before Native American populations diversified in the Americas. The discovery made Siberia look different. 24,000 years ago, the region was not simply a distant eastern edge of Europe or a waiting room for later migrations into the Americas. It was a crossroads where small human populations met, separated, and carried pieces of one another's histories across immense distances. MA1's people had occupied part of that world. Their distinct population did not survive into the present as a separate community. There is no modern nation, language, or living group that can be treated as a simple continuation of the people represented by the boy from Malta. Yet their ancestry did not disappear with them. It entered other populations. It traveled through families who would never know them, name of the people from whom that inheritance came. It became part of the deeper ancestry of later communities in the Americas and across Eurasia. That is the payoff of MA1's genome. Ancient DNA did not merely identify one Ice Age child. It recovered a lost population standing between ancient Siberia, Western Eurasia, and the first peoples of the Americas. Until his genome was sequenced, that population had been almost invisible, present in tools and bone fragments, but absent from the human story in any clear genetic form. The child himself left no account of the cold. He did not describe the people who fed him, the hunters who crossed the steppe, or the fires that kept his camp alive through winter. His bones gave archaeologists a place and a date. His DNA revealed a human connection spanning continents. That matters because extinction does not always erase a population in one clean moment. A people can lose its separate identity while parts of its ancestry continue elsewhere. Its language can vanish. Its camps can disappear beneath soil. Its descendants can merge into other groups until no one remains who carries the old identity alone. But a genome may still preserve the outline. At first, ancient DNA seemed to restore individual lives. One girl, one jawbone, one child. The deeper pattern is harsher. These molecules are beginning to reveal whole populations that vanished from the landscapes where they once belonged. The next trace did not come from a grave at all. It came from an object that held the DNA of a woman long after her own body had disappeared. At first, ancient DNA appeared to revive people through their bones. The deeper question was whether a human trace could survive after the body itself was gone. The next evidence came from something far more intimate, an object worn close to someone's skin. Number 5, the pendant that kept a woman's DNA. It began with a deer tooth, no larger than a thumb, drilled through and shaped into a pendant. Someone carried it through Ice Age Siberia. Roughly 19,000 to 25,000 years later, it was recovered from Denisova cave. No human skeleton lay beside it. No burial told archaeologists who had made it, worn it, or passed it on. Then the pendant gave up human DNA. That was the contradiction. A person's bones may disappear completely, while an object held against the skin can preserve enough biological material to identify the person most closely associated with it. How could a deer tooth retain a human trace after tens of thousands of years, when the human body itself was gone? In 2023, Elena Essel and her colleagues developed a non-destructive method to draw DNA gradually from ancient bone and tooth artifacts. The challenge was obvious. Paleolithic ornaments are rare, while conventional DNA extraction can require drilling into them or reducing part of them to powder. That would damage the very object archaeologists were trying to understand. The team washed the pendant and used a sodium phosphate solution. DNA, released from deeper inside the tooth, could be separated from contamination closer to the surface. The process was designed to preserve the pendant's shape and surface while testing what had become trapped within it. Contamination was the central danger. An artifact may have been touched by archaeologists, conservators, museum staff, and visitors. Modern DNA can overwhelm the older signal. The Denisova pendant was different because it had been excavated and handled with precautions intended to reduce modern contamination. When the recovered molecules were tested, they carried the characteristic damage patterns of ancient DNA. Mitochondrial and nuclear DNA pointed in the same direction. The presumed maker or wearer was female, and her genome had strong affinities with ancient North Eurasian individuals previously found farther east in Siberia. That result did not give the woman a name. It did not prove that she alone made the pendant, wore it every day, or kept it for the rest of her life. The object may have been traded, inherited, handled by several people, or carried through more than one camp. DNA can identify a close biological association. It cannot reconstruct every hand that touched the tooth. Even so, the evidence answered part of the question. The pendant carried the genetic trace of a woman affiliated with an ancient, North Eurasian-related population that no longer survives as a separate community. It extended the known presence of ancient North Eurasian-related people toward Denisova cave and connected one of them to an object of personal adornment. Paleolithic archaeology usually has to keep people and objects apart. A burial may preserve a skeleton, but reveal little about daily life. A pendant may preserve craft, style, and symbolism, yet offer no clear human owner. Here, the two records came together. A tooth taken from an animal became a fragile record of a human being whose own bones may never be found. The human consequence is quieter than a skull or a burial, but it matters no less. This woman lived after Neanderthals and Denisovans had disappeared as distinct populations. She belonged to later modern humans, moving through the same Siberian landscape, carrying a genetic history tied to a population that had already become difficult to see. The clear payoff is that ancient DNA can reveal vanished people through objects they used not only through their bodies or the sediment beneath them. It gives archaeology a new way to connect biological ancestry with the personal material world of the Ice Age. The limits remain real. One pendant cannot stand for every person near Denisova cave, and it cannot tell us what the ornament meant to the woman associated with it. Yet it closes a gap that once seemed permanent. Archaeologists recovered the object. Genetics recovered part of the person. The next evidence file leaves Siberia for an island landscape east of Asia, where the inner ear bone of a young woman revealed a human lineage that no living population carries in the same form. Number 4. The woman whose lineage faded from the islands. In a limestone cave on Sulawesi, a young woman lay on her side with her legs drawn up. Large cobbles partly covered her body. More than 7,000 years later, her skull was crushed, her skeleton was fragmented, and tropical heat had done what it usually does to ancient DNA, almost erased it. One dense piece of bone survived. It came from the petrous portion of her temporal bone, close to the inner ear. From that fragment, researchers recovered the first ancient human genome sequenced from Wallacea, the island zone between mainland Southeast Asia and Australia-New Guinea. The woman was about 17 or 18 when she died. She was buried at Liang Panning in South Sulawesi around 7,300 to 7,200 years ago, within a hunter-gatherer culture archaeologists call Tawalian. Her remains were exposed during excavations that began in 2015. She is now known as Bessé. Her genome did not fit cleanly into any living population in the region. How could someone buried on an Indonesian island 7,000 years ago belong to a human lineage no living community carries in the same form? The first answer came through comparison. Bessé shared much of her genetic history with present-day Papuan and indigenous Australian populations. Wallacea lies between Asia and Sahul, the Ice Age landmass that joined Australia and New Guinea. People moving through these islands had to cross open water, keep small groups alive after each crossing, and build lives on landscapes divided by sea. Yet Bessé was not simply an early member of a population still visible today. Her genome occupied its own position. It represented a previously unknown ancestry profile that branched away around the time Papuan and indigenous Australian populations separated from one another, roughly 37,000 years ago. The analysis also found Denisovan-related ancestry and a deep Asian-related component that did not fit the older, simpler model of the region's population history. The islands were more than a corridor crossed once on the way toward Sahul. They were a meeting ground where different human populations moved, mixed, and sometimes disappeared before later communities took their place. Bessé belonged to a Tolian-associated community that lived thousands of years after the first Homo sapiens reached the wider region. Yet her genome still carried a record of older movements and contacts. It suggested that the history of these islands was not one migration followed by a settled population, but a series of arrivals, separations, and genetic mixtures whose traces were later obscured. What happened to her people? The DNA cannot answer that cleanly. One genome cannot stand for every person who lived within the Toalian cultural tradition. Genetics cannot tell us which language Bessé spoke, what her community called itself, or whether later groups replaced, absorbed, or gradually outnumbered the population to which she belonged. The study supports a narrower conclusion. No present-day population sampled so far carries Bessé's ancestry profile in the same form. Later population movements, including the Austronesian expansion, probably helped reshape Wallaceia's genetic landscape. But the route from her world to the one that followed remains incomplete. That is the clear payoff of her inner ear bone. Ancient DNA revealed a human lineage that had faded so thoroughly from the islands that no living population can simply be named as its continuation. Without Bessé, the Toalians would have remained mostly stone points, scrapers, animal bones, and a culture without a genetic face. Her burial makes the loss feel personal. Someone placed her in the ground, surrounded by material traces of a life that belonged to a human world, now partly erased. She did not vanish from history because she left nothing behind. Until her DNA was read, no one knew what her surviving bone could still say. The next case goes much deeper in time, from the inner ear of a young woman to a thigh bone in Spain, nearly 500,000 years old, one whose maternal DNA appeared to point toward the wrong side of Eurasia. Number 3. The bone that pointed to the wrong family. At the bottom of a deep chamber in northern Spain, archaeologists found a 430,000-year-old thigh bone among thousands of human fragments. The skulls around it carried features long associated with the Neanderthal line. Then, DNA from that bone seemed to point somewhere else entirely, toward Denisovans, a population first identified in Siberia. The site is called Sima de los Huesos, the pit of bones, inside the Atapuerca cave system. Its fossils came from at least 24 individuals, adults and younger people, whose remains accumulated far from daylight. Researchers still debate how so many bodies reached the chamber. Natural processes, deliberate deposition, and other explanations have all been considered. The cave preserved the bones. It did not preserve the decisions that brought them there. For years, their anatomy seemed to fit an old expectation. Their faces projected forward through the middle. Their brow ridges were heavy. Their teeth carried traits associated with Neanderthals. They looked like people standing early on the path toward the later Neanderthals of Europe. In 2013, a team led by Matthias Meyer recovered an almost complete mitochondrial genome from powder taken from one of those ancient thigh bones. Mitochondrial DNA is inherited through the maternal line. It follows only one thread through a family's past, but that thread can still be revealing. The result was difficult to fit into the familiar story. Instead of grouping most closely with the mitochondrial DNA of later Neanderthals, the sequence was more closely related to the lineage leading to Denisovan mitochondrial genomes. The people in the pit of bones lived in Spain. Denisovans were known from Asia. Yet the maternal DNA of this Spanish population seemed to reach across Eurasia toward a group that should have been on the other side of the map. How could people whose bones looked Neanderthal carry a maternal genetic history that seemed closer to Denisovans? That question mattered because it challenged a clean version of human evolution. The older diagram placed ancient Europeans on a fairly direct road toward Neanderthals, while Denisovans formed a separate eastern branch. Sima de los Huesos suggested that the paths had not been so neatly divided. The first genetic result did not prove that Denisovans had lived in Spain. It revealed something more subtle, and in some ways, more unsettling. A maternal lineage can survive inside a population long, after the larger population history has shifted around it. One line of ancestry may retain an older signal, while the rest of the genome records a different relationship. 3 years later, researchers recovered nuclear DNA from 2 Sima specimens. Nuclear DNA comes from both parents and carries a much broader record of ancestry than mitochondrial DNA alone. This time, the result was clearer. The Sima people were more closely related to Neanderthals than to Denisovans, and the split between the Neanderthal and Denisovan populations had already occurred before 430,000 years ago. That was the payoff. The people in the pit of bones were not a hidden Denisovan colony in ancient Spain. They belonged on the Neanderthal side of the family tree. But their mitochondrial DNA preserved an older and more complicated history than later Neanderthal genomes had led researchers to expect. Why did those two genetic records point in different directions? Researchers have proposed several possibilities. Later gene flow may have changed the mitochondrial line, carried by Neanderthals after the Sima people lived. Another possibility is that the Sima maternal lineage preserved an older, genetic relationship that later disappeared from most Neanderthals. The available DNA narrows the possibilities, but it does not settle the exact sequence of events. That uncertainty is not a weakness in the discovery. It is the reason the discovery matters. A single bone showed that ancient human populations did not move through time as isolated branches, neatly splitting and never touching again. Their histories were shaped by migration, separation, contact, and genetic lines that could persist in one population while disappearing from another. The people of Sima de los Huesos had been gone for nearly 500,000 years before anyone read even a fragment of their DNA. Their faces suggested one ancestry. Their maternal genome suggested another. Their nuclear DNA finally revealed the deeper pattern. A population already close to the Neanderthal line, carrying evidence that the human family tree was far more tangled than its simplest diagrams allowed. The next case moves from an ancient population whose genetic history grew more complicated with every test to one of the earliest modern human genomes in Europe. A woman whose lineage entered the continent early, then nearly vanished from the people who came after. Number 2. The woman Europe did not keep. In a cave beneath a hill called Zlatý Kůň, Golden Horse, in Czechia, a human skull lay forgotten by science for decades. Found in the early 1950s, it was repaired with animal-based glues, then dated so uncertainly that no one could agree where its owner belonged in Europe's long human story. DNA from the skull changed that. The woman had lived more than 45,000 years ago, near the beginning of modern human life in Europe. Her genome carried a harder message. She belonged to a population that arrived early, lived in a continent still shared with Neanderthals, and then left no clear genetic continuation in later Europeans or Asians. Why did one of Europe's earliest modern human populations almost disappear from the ancestry of everyone who followed? The woman is known to researchers as Zlatý Kůň. Her remains came from the Koneprusi cave system in central Bohemia. Earlier radiocarbon tests had been compromised by conservation materials used after excavation. The skull had survived. Its date had not. In 2021, a team led by K. Proofer recovered a genome from the bone and found another way to estimate her age. People inherit Neanderthal DNA in segments that become shorter with every generation. Zlatý Kůň carried about 3% Neanderthal ancestry. Her segments were longer than those in the roughly 45,000-year-old Ust-Ishim individual from Siberia. That placed her nearer the original mixing between early modern humans and Neanderthals, making her likely more than 45,000 years old. The deeper surprise lay in where she sat on the family tree. Zlatý Kůň belonged to a population that formed before the ancestors of present-day Europeans and East Asians separated from one another. She was neither an early European nor an early East Asian in the modern sense. Her people represented an older branch of the expansion out of Africa, entering Eurasia before the population patterns familiar today had taken shape. Her skull, therefore, did not mark the start of a line leading toward modern Europe. Genetic comparisons found no clear evidence that her population contributed substantially to later Europeans or Asians. She was part of an early migration that reached the continent, survived long enough to leave a skull in a cave, and then faded from the genetic record. What happened to those people? The genome cannot supply one clean answer. They may have been absorbed into later groups, become too few to detect, moved elsewhere, died out locally, or been overwhelmed by later population changes. The DNA tells us something narrower. Their distinctive lineage did not persist as a recognizable thread in later populations of Europe and Asia. A population can disappear without a single catastrophe. Camps split. Other groups arrive. Families join new communities, and old identities disappear before the ancestry itself is fully gone. In 2024, researchers compared Zlatý Kůň's genome with people who lived around 45,000 years ago at Raunus, Germany. They found distant family relationships. The Czech woman and the Raunus people belonged to the same small, isolated population, one of the deepest known branches of ancestry carried by people who left Africa. That gave the skull a human scale. Zlatý Kůň was not a solitary traveler. She belonged to a connected community moving through Central Europe, while Neanderthals still occupied parts of the continent. The community had relatives across at least 2 regions, yet it left almost no obvious descendants in the people who came later. The second payoff is difficult to overlook. Ancient DNA did not simply age the skull. It recovered an early modern human population that entered Europe before the familiar European story began, then largely disappeared from the genetic future of the continent. The evidence also complicates the old picture of Europe becoming modern human in one steady wave. Several groups entered. Some met Neanderthals and left recent traces of those encounters. Others, like Zlatý Kůň's population, carried older Neanderthal ancestry without signs of later contact. Some became part of later people. Others became almost invisible. A woman died in central Bohemia more than 45,000 years ago. Her bones were repaired, misdated, stored, and nearly lost to the larger story. Then, molecules inside the bone revealed that she had belonged to a human population Europe did not keep. The final evidence file takes that question to its widest scale. A child's finger from Siberia that revealed an extinct human population no one had known existed at all. Number 1. The finger bone that named a lost people. It was the tip of a child's little finger, no longer than a thumbnail, lifted from the cold sediment of Denisova cave in Siberia's Altai Mountains. At first, a fragment that small seemed likely to answer only one basic question: human or animal? No face survived with it, no complete skeleton, no grave. No tools could be assigned to its owner with confidence. Yet the bone carried a genome from a people no one had known existed. The fragment was excavated in 2008 from a layer of the cave that had already produced traces of Neanderthals and later modern humans. 2 years later, researchers sequenced its mitochondrial DNA. The result did not fit either group. The genetic distance was too deep. Whatever child had left that small finger behind, she belonged to a human population missing from every familiar map of prehistory. The question was immediate. How could an entire human people disappear so thoroughly that a child's finger became the first proof they had ever existed? The first answer came from the nuclear genome. Mitochondrial DNA follows one maternal line. Nuclear DNA contains a much broader record, inherited from both parents. When researchers compared the child's nuclear DNA with genomes from Neanderthals and present-day humans, the pattern became clearer. Her people were closely related to Neanderthals, but they were not Neanderthals. They represented a sister population that had separated from the Neanderthal line hundreds of thousands of years earlier. Researchers gave them a name based on the cave: Denisovans. That was the first payoff. Ancient DNA had not merely added a new individual to a known family. It had revealed an entire human population that the fossil record had almost failed to preserve. For decades, human evolution had been told through bodies. A skull carried a brow ridge. A tooth suggested a lineage. A pelvis, a jaw, or the shape of a wrist offered a clue about a vanished person. Denisovans entered the story differently. Before anyone could draw their face with confidence, DNA had already shown that they existed. The cave later yielded more pieces: a few large molars, another small bone fragment, and genetic traces preserved in sediment showed that Denisovans were not a laboratory accident or a single unusual child. They had used the cave across different periods of the Pleistocene, yet their physical remains remained astonishingly scarce. Even now, the bones and teeth known with confidence can be held in a small box. That scarcity makes the next result more striking. The Denisovan genome did not end in Denisova cave. When researchers examined DNA from living populations, they found that some people in Melanesia carry roughly 4 to 6% ancestry from Denisovan-related populations. Smaller amounts appear in other populations across Asia and Oceania. The exact history is complex, and the genetic traces likely reflect more than one episode of contact. But the broad conclusion is secure. Denisovans did not disappear without descendants. How did a population known from so little bone become part of the ancestry of people living thousands of miles from the cave where it was first identified. The evidence points to ancient contact between modern humans and Denisovan-related populations somewhere in Asia. It does not identify one meeting place, one family, or one moment. It cannot tell us what language was spoken, whether the meetings were peaceful, or how the people involved understood one another. DNA identifies biological connection, not the full social history around it. Still, the implication is difficult to avoid. Denisovans were neither a footnote nor a dead end. They were part of a human world in which populations moved, met, separated, and sometimes became family. Their distinct population disappeared. Part of their ancestry did not. That changes the meaning of extinction. Extinction is often imagined as a final door closing. One group is here, then gone. In the ancient human record, the ending was sometimes less clean. A population could vanish as a separate people, while parts of its genetic history continued inside communities that no longer knew its name. Denisova Cave preserves that contradiction in the smallest possible form: a child's finger from a population nobody had named, and a genetic legacy still carried by people alive today. The bone cannot tell us what the child looked like, what she was called, or whether she ever saw another human group beyond the cave. It cannot recover a song, a burial, or a memory of her family. The molecules restore connection, not a complete life. But they do restore something essential. They show that the human past was never a simple march from one species to the next. It was a crowded landscape of populations that overlapped, mixed, survived in fragments, and sometimes vanished so completely that only DNA could call them back. A finger bone no larger than a thumbnail did not simply name the Denisovans. It forced us to admit that the people we remember are only the people whose traces survived. The 10 discoveries in this investigation lead to one conclusion. Ancient DNA does not return the past intact. It cannot give us every face, every language, or every decision that shaped a vanished human life. What it can do is expose the gaps in the story we once thought we knew. A cave floor can remember people after their bones are gone. A jaw can preserve a relative only a few generations away. A pendant can hold the trace of a woman whose body never survived. And a child's finger can reveal an entire people hidden inside the silence of prehistory. The past was not a clean line of replacement. It was a network of families, migrations, losses, and inheritances carried forward in unexpected places. For another evidence file from the human past, choose the video on screen now and subscribe for the next case.